Hello friends, this video is about seed technology part 2 that contains seed basics and genetic principles. The basic parts of seed are embryo, endosperm and seed coat. Embryo is the baby plant. It consists of plumule, radicle and cotyledon. The embryo is the young multicellular organism before it emerges from the seed. The endosperm is a source of stored food consisting primarily of starches. It is the structure that has been formed to provide nutrition for embryo in germination. The seed coat consists of one or more protective layers that encase the seed. It is tough and waterproof, covers and protects the embryo and food supply. Coming to types of seeds. There are two types, one is monocot and one more dicot. Monocot means embryo with single cotyledon, dicots with two cotyledons. In monocots, these are usually endospermic and dicots are non-endospermic except castor seed. Monocots where germination is hypogeal except in onion, and in dicots it is epigeal except in pea and gram. The monocot flower parts are multiple of 3 and in dicots these are multiple of 4 or 5. In monocots, major leaf veins are parallel, in dicots reticulated. Monocots stem vascular bundles are scattered, in dicots these are ring shaped. In monocots, roots are adventitious and dicots it is taproot system. Roots develop from radicle. In monocots, secondary growth is absent, but in dicots, secondary growth is often present. Monocot grains are wheat, corn, rice, millet, sugarcane, banana, ginger, onion, grass. Legumes are pea, bean, lentil, and peanut, then mint, lettuce, tomato, oak, etc. are dicots. Here there is a picture showing the dicot and monocot kind of flower and different plant parts. First, let us see some of the terminologies. Pure seed. It shall refer to the species stated by the sender or found to predominant in the test and shall include all botanical varieties and cultivars of that species. Other seed. This shall include seeds and seed-like structures of any plant species other than pure seed. Species purity. When it is desirable to avoid contamination of one crop species by another similar type, a larger sample is examined and the number of seeds of the species is counted. The result is then expressed as the number of seeds in the weight seed examined, example 2 per kg. Obnoxious weed. There are some species of weeds which are not universally present on all farm and which when established are difficult to eradicate. Weeds of this kind are described as abnormal or obnoxious weeds. Certified seed should be free from them. It is expressed by number in the weight of seed examined. Inert matter. Pieces of broken or damaged seed, one half of the original size or less, straw, chaff, stone, dust, nematode, gall, dead or living units, ergots, etc. That is, materials which have no life and which are not considered as seed of any plants are separated as inert matter and percent by weight is calculated. Here I have given a difference between seed and grain. Seed is any entity. To call it a seed, it should be viable, but grain, it is not viable. Seed should have maximum genetic and physical purity, but it is not required in grain. Seed should satisfy minimum seed certification standards, but it is not required in grain. Seeds are treated with pesticide or fungicide to protect seed against storage pests and fungi, but in grain it is not required or not recommended because it is consumed. Respiration rate and other physiological and biological processes should be kept at low level during storage in seeds, but not in grains. In seeds, production is technically organized but not in grain. Seed should satisfy all the seed quality attributes, but not in grains. Difference between formal and informal system of seed production. Informal system, this is centrally planned system with mechanized production and homogeneous in nature, whereas informal is locally planned unmechanized production using local resources, no specialization, heterogeneous in space and time. Formal seed production exists a system of quality control used to identify and notify varieties, certified and truthfully labeled seeds. In case of informal, these are traditional system of seed processing, use of local seed types, truthfully labeling and other unlabeled seeds. In formal system, seed production is done by national government agencies, state government agencies, government assisted and other cooperatives, MNCs and TNCs, domestic private sector and joint ventures. In informal system, farmer saved seed, farmer to farmer exchange, farmer cooperatives, community groups, non-governmental organizations, seed growers association. In formal seed production, large quantity of seeds are marketed through government-owned companies, private companies and state universities. In informal, small quantity of seeds are marketed through community level. Highly localized use of conventional and unconventional exchange mechanisms. Coming to scientifically produced seed and grains used as seed. In the previous slide, I have differentiated the seeds and grains where I told that grains are not at all utilized as seeds. It is not so. Grains can be used when they have certain properties. Let us see those properties. The scientifically planned or produced seeds, the production of these seeds follow well-designed seed program. These are the varietal purity of this seed is clearly identified from its breeder seed and is genetically pure. During seed production process, quality standards like removal of off-types, disease plants, weed plants and other crop plants are carried out. Seeds have physical purity and good germination. Scientific seed production follow the processing, treatment, packaging and labeling procedures. Drying of seeds is done in a controlled condition. During seed storage, viability and vigor of seeds are maintained. Seeds produced are certified and labeled properly and never be converted into grains unless it is directed by the seed inspector. These all are the properties of scientifically produced seeds. Whereas grains used as seeds has no designed seed program for production, varietal purity of grain in seed is not known, quality standards are not followed, physical purity and germination is not ensured, processing, labeling and tagging are not followed in grain production. Grains are dried in higher temperature and thus quality of seeds are affected. During storage, grains are protected against pests and diseases and not for vigor and viability. Grains which are used as seeds will not have any certification labels and tags, and this can be utilized for commercial grain purpose and sometimes can be utilized for seed purpose. Here there is a table suggesting seed replacement and seed multiplication ratio for different crops. In paddy, The replacement rate is 17 and multiplication rate is 1:80. In pearl millet, replacement rate is 8 and multiplication rate is 1:200. In maize, replacement rate is 6 and multiplication rate is 1:80. Red gram, replacement rate is 6.1 and multiplication rate 1:100. Black gram, replacement rate is 17.7 and multiplication rate is 1:40. Green gram, replacement rate is 11.7 and replacement rate is 1:40. In cowpea, replacement rate is 14.2 and multiplication rate 1:40. In groundnut, replacement rate is 5 and multiplication rate is 1:8. In sunflower, replacement rate is 50 and multiplication rate is 1:50. In sesame, replacement rate is 15 and multiplication rate is 1:250. Coming to the isolation distance. The isolation distance differ in self-pollinated, cross-pollinated and often cross-pollinated crops. In self-pollinated crops, almost all crops have 3 meter isolation distance except black gram and green gram, which has 10 meters. In cross-pollinated crops, maize has 400, pearl millet hybrid 1000, sunflower 400. In often cross-pollinated, red gram is 200 and sesame 100 meters. Germination in plant. Emergence and development from the seed embryo of those structures which seed indicate the ability to produce a normal plant. Conditions necessary for germination are water, air, temperature, light, etc. The modes of germination are: seeds present in 2 modes of germination based on the behavior of cotyledons or storage organs. 2 types of mode of germination are epigeal and hypogeal germination. Epigeal germination. The cotyledons come out above the soil surface and generally turn green and acts as first foliage leaves. This type of germination is present in groundnut, beans, cotton, sunflower, cotton, etc. Here in this picture you can see the epigeal kind of germination, whereas in dicots the seeds come out of the soil surface and they get germination, and after a period of certain growth the cotyledons will shred off. Hypogeal germination. Here the cotyledons do not come above the soil surface. This type of germination is found in wheat, barley, maize and pea. Coming to principles of seed production. General principles. Production of genetically pure and otherwise good quality pedigree seed is an exciting task requiring high technical skills and comparatively heavy financial investment. During seed production, strict attention must be given to the maintenance of genetic purity and other qualities of seeds in order to exploit the full dividends sought to be obtained by introduction of new superior crop plant varieties. In other words, seed production must be carried out under standardized and well organized conditions. Agronomic principles. Selection of a agroclimatic region. Crop variety to be grown for seed production in an area must be adapted to the photoperiod and temperature conditions prevailing in that area. Selection of seed plot. The plot selected for seed crop must be free from volunteer plants, weed plants and have good soil texture and fertility. The soil of the seed plot should be comparatively free from soil-borne diseases and insect pests. Isolation of seed crops. Isolation from other nearby fields of the same crop and other contaminating crops as per requirement of the certification standards. Preparation of land. Good land preparation helps in improving germination, good stand establishment, and destruction of potential weeds. It aids in water management and good uniform irrigation. Selection of varieties. Variety of seed production must be carefully selected, should possess disease resistance, earliness, grain quality, high yielder and adapted to agroclimatic conditions of the region. Seed treatment. Depending upon the requirement, seed treatments are given. First one is chemical, second one is bacterial inoculation for the legumes like rhizobium treatment, then seed treatment for breaking dormancy. Time of planting. The seed crops should invariably be sown at their normal planting time. Depending upon the incidence of disease and pest, some adjustments could be made if necessary. Seed rate. Lower seed rates than usual for raising commercial crops are desirable because they facilitate roguing operations and inspections of seed crops. Method of sowing. Most efficient and ideal method is by mechanical drilling. Depth of sowing. This is extremely important in ensuring good plant stand. Small seeds should usually be planted shallow, but large seeds could be planted a little deeper. Roguing. Adequate and timely roguing is important. It must be of the field crops, may be done at any of the three stages. One is vegetative or pre-flowering stage, during flowering stage, or at the maturity stage. Supplementary pollination. Provision of honeybees in hives in close proximity to the seed fields of crops largely cross-pollinated by the insects ensure good seed set, thereby greatly increase seed yields. Weed control. Good weed control is the basic requirement in producing good quality seed. Weed may cause contamination of seed crop in addition to reduction in yield. Disease and insect control. Successful disease and insect control is another important factor for raising healthy seed crop. Apart from reduction of yield, quality of seeds from diseased and insect damaged plants is invariably poor. Nutrition. In the nutrition of seed crops, nitrogen, phosphorus, potassium and several other elements plays an important role for proper development of plants and seed. It is thereby advisable to know and identify the nutritional requirement of seed crops and apply adequate fertilizers. Irrigation. It can be important at planting for seed crops on dry soils to ensure good uniform germination and adequate crop stands. Excess moisture or prolonged drought adversely affects germination and frequently result in poor crop stand. Harvest of seed crops. It is of great importance to harvest a seed crop at the time that will allow both the maximum yield and best quality seed. Drying of seeds. In order to preserve seed viability and vigor, it is necessary to dry seeds to save moisture content levels. Storage of raw seeds. The best method of sowing seed for short periods is in sacks or bags in ordinary buildings or godowns. Coming to the maintenance of genetic purity during seed production. Various steps to maintain purity are using approved seed only in seed multiplication, inspection and approval of field prior to planting, field inspection and approval of growing seeds at critical stages for verification of genetic purity, detection of mixtures, weeds and freedom from noxious weeds and seed-borne diseases, etc., sampling and sealing of cleaned lots, growing of samples of potentially approved stocks for comparing with authentic stocks. Various steps suggested for maintaining genetic purity are providing adequate isolation to prevent contamination by natural crossing or mechanical mixture, roguing, periodic testing of varieties for genetic purity, avoiding genetic shifts by growing crops in areas in their adaptation only, certification of seed crops to maintain genetic purity and adequacy or quality of seed, adopting the generation systems like breeder, foundation and all, then grow out test. Thank you. I hope you liked my video and do not forget to subscribe my channel.